Ever wondered what examiners are really looking for when they're marking your paper? It can feel like a total mystery, right? Well, today, we're pulling back the curtain. We're going to demystify the whole thing by breaking down a pretty complex genetics question from a real past exam.
And we're not just guessing here. Our source for this is the official AQA GCSE Biology Higher Tier Mark Scheme from June 2023. This is the real deal, the exact document examiners were using. You can't get any closer to the source than this.
So this is the big question, isn't it? How do you actually make sure you get every single mark, especially on those trickier questions? That's the goal, after all.
The secret, really, is to start thinking like the person with the red pen. And their rulebook is the mark scheme. So let's dive in and take apart question 8 from this paper piece by piece.
All right, first up is question 8.1. This part was all about putting the stages of a biological process into the correct sequence.
So the original exam paper had a diagram showing 6 stages of meiosis, and they were just labeled A through F. Your job was to get them in the right order. We don't have the pictures in front of us, but the mark scheme tells us the exact sequence they were looking for.
And here it is. The one and only correct answer was A, then E, D, C, B, and finally F. To get that single mark, you had to have it in that exact order. No wiggle room on this one. Okay, let's keep it moving and see how this builds. Question 8.2 shifts gears from the order of events to the numbers, specifically the number of chromosomes.
So the puzzle was this. You had to state the number of chromosomes in the cell right at the start of meiosis, and then the number in each cell at the very end. This is a classic test of a really fundamental concept.
And there you have it, 16 at the start, 8 at the end. This perfectly shows why meiosis is often called a reduction division. It literally reduces the chromosome number by half, getting the cell ready for fertilization. Now this is where it gets really useful. You get 1 mark for saying 16 and a second for saying 8. But look at that extra info. The mark scheme allows 8 pairs for the start number, which shows they're looking for your understanding of how chromosomes are organized. This is like getting a peek inside the examiner's head.
All right, now for the why question. Part 8.3 goes beyond just describing what happens and asks why meiosis even happens in the first place. This is where really understanding the topic pays off, not just memorizing it.
To get the first of the 3 marks available here, the core idea is actually pretty simple. Meiosis happens to form gametes. That's the first step to a perfect answer.
So, how do you grab all 3 marks? You have to connect the dots. First, you say it forms gametes. Second, you explain that 2 gametes fuse during fertilization, and third, you explain the result. This process keeps the chromosome number constant from one generation to the next. And notice the flexibility. Sex cells or eggs and sperm are fine. For the result, prevents doubling also gets the mark. This stuff is absolute gold dust for your exam technique.
Next up, question 8.4 digs into another key result of meiosis, genetic variation. How does this whole process manage to shuffle the genetic deck so effectively? To get both marks here, the mark scheme was looking for a 2-step explanation. First, you had to say that a random chromosome from each pair is involved. That word random is so important, it's the entire source of the genetic shuffling. Then, for the second point, you had to describe where it goes, that it moves to one end of the cell to eventually end up in a new gamete. We're on the home stretch. So we've seen how meiosis shuffles the genetic deck, so it makes sense to look at the code itself. These last 2 parts of question 8 are quick-fire questions on DNA structure. Let's tackle them together. For question 8.5, they pointed to a specific part of a DNA diagram and just asked, what is this thing? A pretty straightforward identification question. And the answer for 1 mark was just the word base or bases. You could also say organic base or nitrogenous base. But check out that last instruction, ignore adenine/cytosine. This tells you that even if you gave a correct example, it was irrelevant here. They wanted the general term.
Okay, now for the very last part, 8.6. This was a calculation based on that same DNA diagram. You had to carefully count a specific component, and if you did it right, the answer was 12.
And here we go again. You can see that built-in flexibility. While 12 was the most direct answer, the mark scheme clearly says that writing 6 pairs, or even spelling it out as 6 pairs, would also get you the mark. It shows they're testing your understanding of the structure, not just your ability to count. So, after all that, what are the big takeaways? Well, first, use precise biological language. Words like gamete or random matter. Second, really understand the why behind the what. And third, remember that there are often different ways to say the right thing, so be confident in your own words if you know the science.
You know, a phrase like this, pulled straight from a mark scheme, might seem a bit dry, but when you think about it, it's a really elegant description of a vital biological process that ensures life can continue, generation after generation. It's pretty amazing, really.
Peeking behind the curtain like this isn't about finding shortcuts. It's about understanding the rules of the game. It lets you show the examiner what you really know. So now that you've had a look inside, what are you going to tackle next?